Good morning, and first let me to uh, apologize for not being there with you, but my schedule for the last months of the year is crazy. So at the same day where you the meeting in Poland will occur, I will be in another meeting in Italy. So uh, we agreed to record a presentation and that will be presented during the meeting. Of course, I hope in the near future will be some other chances to present the exosomes to you there in Poland. Okay, so this presentation is just to present to you the word of exosomes and what we obtained just working on exosome derived from plants. Just briefly to present myself, I'm a professor in medicine and gastroenterological science, and I run in different fields in research in medicine, such as because of immunity, neuroimmunology, virology, and oncology. My most important discovery of mine uh, have been some mechanism underlying giant cell formation in granulomatous disease such as Crohn's disease, some very interesting mechanism of HIV-1 entry and release in infected cells, the connection between the membrane of the cells containing that receptors and the cell cytoskeleton. And I guess the one I was very proud is to build the scientific evidence that tumors may be cured through an anti-acidic therapy. So we discovered some mechanism underlying tumor cannibalism. And last but not least, we studied for 20 years both human and plant-derived exosomes. I've been head of research at the Italian NIH, and there for 10 years, the head of the anti-tumor drug section. Um, and now I am the scientific director of Exolab Italia. We based this innovative startup on and set up new technology to obtain exosome from plants, and these plants derived from the organic agriculture of Italy, and this is the reason why our name was Exolab Italia. Um, our startup was based on research and innovation, and um, because our products are daily um, implemented by new discoveries, and our products are the results of the real discoveries. So we are proud to say that we are between a very few um, startup in worldwide that are able to study new products and produce exosome-based products um, based on plant-derived exosomes that are probably we are the only one providing exosome deriving from organic agriculture, Italian organic agriculture. Um, this is slide is to emphasize what I've said you, and in Italy we have up to 1,500 different plant varieties. This biodiversity is is one of the most recognizing feature of Italy, because Italy is a very special um, country that is a sort of you know pier within the very calm sea, such as the Mediterranean, with a north to south, east to west position that allows a very peculiar biodiversity in our country, and we figured out to obtain exosomes from different plants, meaning different vegetables and fruits deriving from Italian organic agriculture. One thing that needs to be emphasized is that we are an innovative startup with the zero waste, with a high level of sustainability, and this is another very important feature of us, and based on very peculiar exosome-based technology, leading to products that may be used in skincare and food supplementation. Um, However, just after the presentation of our startup, I guess it is important just to give you some information on what exosomes are actually. And exosomes are nanovesicles, vesicles belonging to a family of extracellular vesicles that are of a nanometric size, meaning between 50 to 180 nanometers. Exosomes are released by all the cells, um, you know, from released from all live beings, including plants, humans, and animals, and microorganisms as well. Um, what is very very important is that they are um, the natural delivery systems of all the live beings. And for instance, we know that in human body they connect all the organs compartments between them, um, each wilds of our life. But what is very important concerning the plant-derived exosome is that they connect uh, the different species between them, and some are saying that they are connectors between kingdoms, and this may occur to the air, to the water, to the soil as well. They may have been described for the first time by electron microscopy, and in the picture, you can see a very special electron microscopy technology allowing in an exosome suspension just to recognize single exosomes and to study roughly their size. And you can see in this picture that these are exosomes from some citrus, um, citrus paradise, that is grapefruit, for instance, that are very similar in terms of size. Um, as I said, the exosome belong to a family of extracellular vesicles, meaning that they are vesicles with, uh, in terms of phenotype, the same proteins that you can find on vesicles contained within cells, but you can find them only outside the cells, and also in the all the liquids or the fluids outside the cells. And this is just to give you an idea on the size. Exosomes are, plus or minus of the same size of viruses. And in the same family, we have microvesicles that may range from 150 micrometers to 1 millimeter, and 1 micron, from 0.5 micrometer to 1 micrometer, and they may resemble in terms of size to the vast majority of micro organisms, bacteria, and so on. So in this family also, uh, necrotic bodies, apoptotic bodies are included, but it is an academic inclusion. But when we talk about extracellular vesicle, actually we talk about exosomes and microvesicles. Um, about the mechanism of formation, this is a mechanism shared by all the exosomes from released from cells, either human or animal derived, or plant derived. This mechanism implies a sort of um, fusion, repeated fusion between the internal vesicles, including endosomes, lysosomes, and so on. And when it occurs very close to the plasma membrane, they form these structures that are called multivesicular bodies. They contain the exosomes, and they are beneath the membrane, and they are released, in different condition from this structure to the extracellular environment. This is a mechanism that's shared by all the exosomes and extracellular vesicles as well. Um, how the exosomes may interact with cells, for instance. And between the mechanism most recognized by the all scientists involved in the field, there are two mechanisms. One is involves ligand to receptor interaction. For instance, exosome may express a ligand that may link to to a receptor present on a cell membrane, then triggering an internal pathway of metabolism. And then the other one that is probably the most used by the exosome is the fusion between the exosome membrane and the membrane of the plasma of the cells. This is a mechanism that allows the uploading of the exosome content within a cell. This meaning that this mechanism allows exosome to pass some, barriers that are considered a sort of sacrarium in the past, such as the brain barrier, very easily. But in any event, is the most important mechanism allowing exosome to um, connect cells between them and organs between them, and different spaces between them. We have a very innovative technology platform that allows the preservation of the exosome structure, because it implies not chemical treatments but only physical treatment. Um, in fact, we use two different accepted methods to obtain exosome following, uh, physical extraction from fruits and vegetables. To obtain exosome are needed repeated rounds of centrifugation and ultracentrifugation, and we are studying setting up a new method based on filtration. After that, exosomes are um, lyophilized using sublimation, and then obtaining a pellet provided as first raw material. How we can we analyze our exosome extracted from plants, meaning fruits and vegetables. We use three methods that are quantitative. One is a nanosite tracking analysis. The other one is nanoscale flow cytometry, and the last one set up in our lab is a monocapture based ELISA. But also, we also use two qualitative methods that are electron microscopy, that is a mandatory method to have a look on your sample, and also Western blot that I may say to you, we have these proteins, we have the proteins we are looking for. Um, Let me to show to provide you some details on nanosite tracking analysis. Is very important equipment in our process. That is a method that through a laser recognize the particles that are triggered by a laser ray. The particles that diffuse within the sample are analyzed for their Brownian movements, and through a very important algorithm they are counted and evaluated for the distribution in terms of size. This equipment allows also to analyze the fluorescent particles as well. This slide is also to emphasize how they focused our attention, on plant-derived exosomes. Um, they are very interesting because most of all, um, they have the same size of human exosome. They deliver proteins and lipids. They contains as human exosome DNA, RNA, and microRNA. They are highly fusogenic as the human exosome are, but they have a lot of very important advantages as compared to human exosome because they are very, very bioavailable. They have a phospholipids lipid membrane that allows them to pass the gastric pH and to pass the brain barrier, and you know, and also to serve as a delivery system. But it is very important. They are very very high level of bioavailability. They are not dangerous because the human exosome may contain dangerous molecules such as viruses, but also toxics accumulated during life, and also drugs and so on. And most of all, the the human exosomes are not suitable for industrial production because they are mostly obtained from cells, and also whether you can fill up, a very big hangar with incubators containing human cells, you can't obtain a real scalability for the industrial use. So these are the very real advantages of plant-derived exosomes. Um, but this is a picture, explaining to you much better the way exosomes are released and the way they interact with cells. But I should, I said to you most of what is contained in this slide actually. Um, this is a very important, information for cosmetic use of plant-derived exosomes because they may pass, the the external layers of the skin through three different mechanisms. They may, on one end, allowing to penetrate the exosome along the air as then they fuse through the cells containing in the lower layers of the skin, and they can be pass the skin through a sort of transdermal mechanism. Um, these are the difference between the plant exosome and human exosome and the artificial, nanoparticles that are called liposomes. They are, particles of lipids. As you can say on your left, plant exosome contains all contains nucleic acids coming from nature. They contain secondary metabolites. They are scalable. They have a very high level of biomimetism. They are not non-toxic. They are very efficient in their function. And you can say uh, the human exosome do not contain secondary metabolism. They are non-industrial scalable. They are very, dangerous in terms of toxicity, and the artificial, nanoparticles, have a red light for all the these features. So they have a lot of advantages, and this is the most reason we are focusing and we continue to focus on plant exosomes instead of human exosomes and liposomes. We have two patents of exosome allowing, on one end, talking about, the method we use to obtain exosome, and the other one is a method to upload any molecules within plant exosomes. That meaning other molecules that are not bioactives that are not contained primarily in the exosome we used to in cosmetics or in nutraceutics, but also to upload the exosome with synthetic drugs, chemical drugs, when is needed. Uh, we have a lot of scientific data that I would like to show you, obtained both in vitro and in vivo. Um, First of all, I should say to you that what is very original in the Exo Lab Italian strategy is to use not single fruits to obtain single fruits derived exosomes, but to use mixes of exosomes deriving from different fruits and vegetables that put together may have shown to have a synergic effect. Um, we are talking about, of the exocomplex that will be used by Arkana that contain actually apple, mandarin, and papaya. And we show you very briefly data showing that these exocomplex increase the fibroblast proliferation, reduced cellular duplication, reduced the aging effect on the skin and on the cells, belonging to the skin layers, reduce the mitochondrial oscillation, restore the membrane potential of the mitochondria, and restore C3 1 production that is very important bioactive for the skin, and increase amazingly the wound healing. So just to show with the numbers, first of all, this is the content and then in the average of the exosome, the exocomplex we are using. It contains a lot of antioxidants such as superoxide dismutase, glutathione, catalase, ascorbic acid, but also melatonin, and showing as a whole a very high level of antioxidant capacity. It contains also phenolic compounds, and this is surprising. Also ATP, suggesting that exosome may produce energy. So this is a test on the anti-aging activity, performed on fibroblast, human fibroblast culture. And you see from the, on this test that there was in the fibroblasts treated with the exocomplex, there was an 100% increase of the proliferation um, in terms of optical density. And there is a reduction in duplication. So there are many, many cells with a reduced in duplication time. Uh, you know, More cells with a reducing time, in a reducing time. Then, on the same cells, we tested the cellular metabolism. As you can see, there was a 90% the decrease of accumulation of mitochondrial superoxide within the treated fibroblasts as compared uh, to the fibroblasts submitted to oxidative stress. Then we tested the mitochondrial membrane protection, and you can see that there was an increase of 487% of the mitochondrial membrane protection in uh, fibroblasts undergoing exocomplex stimulation as compared to the fibroblasts undergoing oxidative stress. This is the production of sirtuin, and on the same cells, there was 25% increase of sirtuin production, sirtuin 1 production by treated fibroblast. This is, I don't know whether from this slide you can realize, but actually, this is an in vitro test that is called wound healing, because you have a monolayer of cells that where you can produce a sort of wound, so, sorry, a sort of wound here. This is the time zero, and this is the time, 24 hours time, and 8 hours time following the exocomplex treatment, when you have 60% of filling up of the wound, and the total filling up at 48 hours. This is the vimentin production in the same cells, and you can see an increase, 31% increase in vimentin production in the same cell culture. This is, the increase of um, collagen production in treated fibroblasts. This is the time zero untreated fibroblast, and this is the fibroblast culture following treatment with the exocomplex. You can see the green cytoplasm is the amazing increase of collagen production in treated cells. These are the publication, the scientific publication, where you can see the vast majority of these results. And this one, the last one on the bottom, is the last publication that is relative to the nutraceutics that will be, you know, suggested be implemented in skin treatment by oral treatment. I guess I I don't know whether I've been sufficiently clear and easy to be understood for from your side, but actually it is a very new and challenging area of scientific interest, and we are very proud together to Arkana to have you know to render available our products for cosmetics and nutraceutical use. And after that, I have to say thank you for your attention. And again, I hope to meet you in the near future in order to hear your questions and comments on our work in Exolab Italia.